Welcome to Crumbs.
For the past 2 years, the Beckman Institute at the University of Illinois housed a multi-person, room-sized, three-dimensional virtual reality system known as the Cave. Developed at the U of I's Electronic Visualization Lab, the Cave utilizes a Silicon Graphics Onyx workstation to rear-project computer-generated images onto the front and left walls, as well as project onto the floor. Another computer, a Silicon Graphics Indie workstation, provides the audio signals. Together, the two computers create the illusion of immersion by portraying a constantly changing perspective of sight and sound when a user wearing a location sensor moves within the display boundaries. Simulator controls may be activated through the use of a handheld pointing device called a wand, or most recently, by using voice commands.
Global VR. Grid.
The Biological Imaging Group at the National Center for Supercomputing Applications developed a program for the Cave which allows the tracking of fiber structures within volumetric data sets. This software, called Crumbs, allows a scientist to image a portion of his data while placing markers along the fiber of interest. The Crumbs software will measure the length of the marked path as well as save the location of markers for later analysis. Professor Timothy Carr at the University of Chicago studies the fate of Drosophila sperm following fertilization. His development of anti-sperm antibodies for use with confocal microscopy has allowed him to image sperm tails in 12 species of Drosophila. The extreme length and compaction of the sperm in the egg prevent Professor Carr from measuring its length using a standard workstation environment, so he turned to Crumbs to help him trace the tail in 3 dimensions.
Crumbs allowed us to actually follow this tortuous path in its entirety because we were able to move through the data set at will and in 3 dimensions, so that as the structure became more and more highly entangled and wrapped back around and onto itself, we were actually able to trace a single strand. By dropping a crumb and knowing where we were, and we had a reference point, we were then able to move to the next point. And by continually doing this, and we dropped more than 2,000 of these data points along this twisted path.
Professor Joanne Urell at the U of I's Department of Veterinary Biosciences, along with her student Janet Sinhanlin, recently reconstructed a 48-hour-old chick embryo from serial section light microscopy.
The yellow region right here, this yellow tube, represents the foregut area, outlined in yellow for the lining endoderm. Then up here are 2 dorsal aortas in red here, paired on either side of the midline. The blue structure over here on the left-hand side is the neural tube, and over here on the far right in red is the heart bending around in its tube-like configuration.
Crumbs is used to trace the path of blood flow through the developing heart as the heart transitions from a straight tube into a 4-chambered structure.
Traditionally, students have used 2-dimensional teaching aids, pictures, photographs, microscope slides, and they have had to mentally reconstruct the 3-dimensional embryo from these teaching aids. With Crumbs, we're able to present to them an actual 3-dimensional image of the embryo at different stages in development.
Rachael Brady, a member of the development team at NCSA, explains Crumbs' role in remote instrument control at Supercomputing 95.
In December, we're going to demonstrate how Crumbs has the ability to not only visualize data sets from various kinds of instruments, but also control these instruments remotely. The way we're going to do this is live from the floor of Supercomputing 95 in a cave in San Diego. We are going to talk to 3 microscopes, which are housed in Illinois. These microscopes are the magnetic resonance imaging spectrometer, the electron microscope, and the scanning tunneling microscope, all of which are at the Beckman Institute.